In molecular basis of inheritance, the DNA, the search for genetic material, the RNA world, replication, then transcription, then genetic code, then translation, then regulation of gene expression, then human genome project, then DNA fingerprinting. So in the DNA, the structure of polynucleotide chain, at 5' end it is free phosphate, at 3' end it is 3 hydroxyl OH group, then double helix model for double stranded DNA given by Watson and Crick. Adenine, thymine, guanine, cytosine are constant and equals 1. The ratios between adenine and thymine and guanine and cytosine are constant and equals 1. So this double stranded polynucleotide chain. There are hydrogen bonds between them, base pairs, the DNA double helix. Now central dogma given by Crick. So DNA to DNA it is replication, then DNA to mRNA it is transcription, then mRNA to protein it is translation. Now packaging of DNA helix, you see it is nucleosome. What inside the nucleosome? There is core of histone molecules. Okay, it is histone octamer, and then around that DNA is wrapped. H1 histone is linker between DNA ends. You see there are histones. They are positively charged basic proteins. So these are histones. They form histone octamer that makes a nucleosome. Okay, and then nucleosome forms the chromatin. There are non-histone chromosomal proteins, NHC, that are euchromatin and heterochromatin. Two types of chromatin. Then the search for genetic material transforming principle given by Frederick Griffith in 1928. Okay, S strain injected into mice die, R strain injected into mice live, S strain hit kill injected into mice live, S strain hit kill plus R strain live injected into mice die. Okay, heat tails transformed. Okay, R strain bacteria transformed by heat kill S strain bacteria. Now biochemical characterization of transforming principle given by Oswald Avery, Colin MacLeod, MacLean McCarthy. Okay, now genetic material is DNA. Alfred Hershey and Martha Chase in 1955 unequivocal proof. You see the Hershey Chase experiment. You see bacteriophage that infects bacteriaceae E. coli. Now radioactive 35 S35 labeled protein capsule, radioactive phosphorus 32 labeled DNA, then first step infection, then blending, and then centrifugation, right? You see no radioactivity S35 detected in cells, but radioactive detected in supernatant form, that is S35 outside. But radioactive E32 detected in cells and no radioactivity detected in supernatant form. So this is how radioactive P32 labeled transformed into the cell. Properties of genetic material DNA versus RNA. Now RNA world, you see Watson-Crick model for semi-conservative DNA replication. DNA replicates semi-conservatively, right? Then there is replication, which is semi-conservative. Now the experimental proof. First in E. coli by Michelson and Stahl. You see separation of DNA by centrifugation, Michel and Stahl experiment.